Hello everyone, I'm Janvi Swami. Welcome to the channel. Today we are going to study about blood groups. Definition of blood groups. Blood groups are specific classifications of blood based on the presence or absence of certain proteins, antigens, on the surface of red blood cells, RBCs. These antigens determine compatibility during blood transfusions, organ transplantation, and also have medico-legal importance in paternity testing. Main blood group systems are ABO blood group system, Rh factor system, M and N factors. Other minor systems like P, Lutheran, and Lewis also exist but are less significant clinically. First system is ABO blood group systems, in which concept of agglutinogens and agglutinins are majorly studied. The ABO blood group system is based on the presence of agglutinogens, antigens, on RBCs and agglutinins, antibodies, in plasma. Agglutinogens A and B are the surface markers. Agglutinins A, anti-A, and SS, anti-B, are present in plasma. These determine four major blood groups, A, B, AB, and O. This table shows about different blood groups, antigen on RBC, antibody in plasma, and which blood group can receive blood from which blood groups. Note is there with respect to blood groups table. These, universal donor, and recipient, labels are only applicable within the ABO system. The Rh factor can change compatibility outcomes. Formation and characteristics of agglutinogens. Agglutinogens start appearing by the 6th week of fetal life and reach adult levels by puberty. They are polysaccharide molecules found not just in red cells but also in organs like the liver, pancreas, salivary glands, and testes. Humans are divided into four main groups. A, B, AB, and O subgroups also exist. A1, A2, A1b, and A2b, with A1 and A1b being more common. The inheritance pattern follows Mendelian genetics with three allelic genes A, B, and O. Second blood system is Rh factor system. The Rh factor, rhesus factor, is another important antigen group discovered by Landsteiner and Wiener in 1940. About 85% of people are Rh positive, Rh+, meaning their RBCs contain the D antigen. Those lacking this antigen are Rh negative, Rh. The Rh system includes 6 main antigens, C, D, E. Among them, D is the most significant, as it strongly triggers antibody production. Rh incompatibility and its clinical importance are blood transfusion reaction and erythroblastosis fetalis, hemolytic disease of the newborn. Blood transfusion reaction like, if Rh plus blood is transfused into an Rh person, the recipient's immune system forms anti-Rh antibodies. On a second transfusion, these antibodies destroy donor RBCs, causing dangerous hemolytic reactions. Hence, Rh testing is essential before any transfusion. Erythroblastosis fetalis, hemolytic disease of the newborn, occurs when an Rh mother carries an Rh plus fetus. The fetal Rh antigen can enter maternal circulation, causing her to produce anti-Rh antibodies. These antibodies cross the placenta and destroy fetal RBCs in later pregnancies. The baby may develop severe anemia, jaundice, or even die if untreated. Prevention is there with respect to Rh factor. Rh-negative mothers are given anti-D immunoglobulin, Rho-D, injection, soon after delivery or miscarriage to prevent antibody formation. Rh agglutinogens and antibodies are like. Every person carries 3 Rh antigens, one from each pair. C, D, E. Dominant genes, C, D, E, make a person Rh+, while recessive genes, C, D, E, make them Rh. The anti-D antibody is the most clinically important because it causes strong immune reactions. Two forms of anti-D antibodies are complete, saline, antibodies. Act immediately in saline or albumin medium, and incomplete antibodies. Act only in special conditions, albumin or Kuhn's test. Third type of blood system M and N factors, medico-legal significance are. The M and N blood group system is another inherited blood group identified by Landsteiner and Levine. It has 3 types, M, N, and Mn. Though not important for blood transfusions, it is used in paternity testing and forensic studies. Each person carries 2 blood genes, M plus M group M, N plus N group N, M plus N group Mn. If a baby's blood group is M, both parents must carry at least 1 M gene. Similarly, Mn combinations can help determine if a suspected person might or might not be the biological father. The blood group substances are nitrogenous heteropolysaccharides containing D-galactose, fucose, glucosamine, and galactosamine. Last type of blood system is P system. Another minor group discovered by Landsteiner and Levine, 1927, is the P system, P plus and P. When these investigators found a new antigen present on the erythrocytes of about 80% of individuals, who were type P, plus, and absent on others, who were typed P. The system was defined by one antibody, anti-P, and one antigen, P. It's not clinically significant but adds to the genetic understanding of blood classification. What is the P-null blood group? The P-null blood group is an extremely rare blood type within the P blood group system. Individuals with this blood group lack all P system antigens, P, P1, and PK, on their red blood cells. Okay, thank you guys for listening with patience. See you on next topic.